Alright, continuing our journey through biomolecules, we are now going to discuss lipids. So there's 3 different kinds of lipids to go through, and we are going to discuss the structure and function of each. The first one that I want to talk about is triglycerides. They're named because they have glycerol and they have 3 fatty acids. Glycerol is this 3-carbon triol that you see, and the way that it's put together with the fatty acids is dehydration synthesis. And we've discussed dehydration synthesis before with proteins. It is also involved in making the phosphodiester bonds in DNA. And now we're seeing it in lipids. So we just take this OH and this H, and out comes water, and we make a new bond. We take this OH and this H, out comes water, we make a new bond, and a third time. And you might note that for the first time, I am not really using all the atoms, like the carbon and hydrogen. I'm just drawing the skeletons. It means the same thing. It just means a long hydrocarbon. These fatty acids that I've drawn here, these are saturated fatty acids. And saturated means that they're saturated with hydrogens. So there are no double bonds, there are no cycles, there is nothing that you would need to take out a hydrogen in order to create. If I started to put double bonds here like this, these would become unsaturated fatty acids. And the reason that this matters is that the geometry of double bonds, they make kinks in the fatty acids, and then if you have a bunch of lipids together, it will be more fluid if it's unsaturated. And if it's saturated, like if there are no double bonds, then everything can fit very close together and it becomes more solid. So for example, butter is a saturated fat, and then something like oil is unsaturated. These triglycerides are what we think of when we stereotypically think of fat. We eat a lot, it gets stored in these triglycerides in the form of these really long carbon chains that have a lot of chemical potential energy. The next one that we're going to talk about is the most important one. That's the phospholipid. The phospholipid makes up our cell membranes, and basically it's like the triglyceride, except you take off one of the fatty acids and you replace it with a phosphate group. This makes a very interesting combination because when you have fats, they are what we call hydrophobic or water-fearing. So all those carbons and hydrogens that have similar electronegativity, they don't have the capacity to do dipole-dipole forces or hydrogen bonding or any of that. So they don't really like to hang around with water that's very, very polar. But here we have the phosphate group, and the phosphate group is polar. So it's actually hydrophilic. So we've got a hydrophilic head, that's the phosphate, and hydrophobic tails hanging off the end, and those are the fatty acids. The way this works in our cell membranes is we end up with a phospholipid bilayer, where all the hydrophobic heads of these phospholipids go on the outside, and they can interact, the hydrophilic heads, not hydrophobic, they can interact with the water on the outside. Meanwhile, the hydrophobic tails, the fatty acids, are going to be on the inside of the membrane. This makes it hard for a lot of stuff to cross the membrane because there is both hydrophilic and hydrophobic stuff in the way, and that's how the cell protects itself. If you look at this diagram of the cell membrane, you'll notice it's not just the phospholipids. You're going to have proteins in there too, and like some glycoproteins or proteoglycans, which are like protein and sugar combined. You're also going to have cholesterol in there. We're going to talk about cholesterol in a minute. It's a steroid. But anyway, you're going to get questions probably about proteins that are embedded in the membrane, and the middle part of them has to be made up of hydrophobic amino acids because it's with the hydrophobic fatty acid tails, and the outsides of them on either side of the membrane have to be hydrophilic because they are surrounded by water. So this last class of lipids we're going over is called steroids, and I know when we think of steroids we think of like the drugs that athletes take to get all buff and it's illegal, but steroids are actually very important in our bodies. They are based on this 4-fused ring structure, and a lot of them are derived from cholesterol. Cholesterol is used in our cell membranes to help them have the right amount of fluidity so that everything in the membrane can kind of move around the right amount. But cholesterol can make, if it's varied a little bit, it can make stuff like estradiol, which is a form of estrogen, or testosterone, or other hydrophobic hormones. And the cool thing about these hormones is because they're hydrophobic, they can actually pass right through that middle of the cell membrane, and they can go and diffuse into the cell and help out with signaling. So the way that these hormones work is they diffuse straight through that membrane, and they're going to complex with some kind of protein inside the cell. And this complex together is known as a transcription factor. And what that transcription factor is going to do is it's going to bind to DNA, and it's going to bind to DNA here, and it's going to attract over RNA polymerase that makes RNA from DNA. And that's going to stimulate transcription and translation. So all of a sudden, this transcription factor, this complex of the hormone and whatever other receptor it's with, is going to say, hey, gene, you need to make your product a whole bunch. And that's going to be the response to the hormone. So those are the 3 kinds of lipids. I hope this helped. Next time we're doing sugars.